Hello, and welcome back to the Youth for Neuro podcast. My name is Pranali, and I am a high school junior and your host for this episode. I'm joined today by Dr. Lisa M. Monteggia, an American neuroscientist who is a professor in the Department of Pharmacology, Psychiatry, and Psychology as well, and is the Barlow Family Director of the Vanderbilt Brain Institute at Vanderbilt University. To provide some background, Dr. Monteggia's educational training began with a BS in microbiology and a master's in biology at UIUC. She then attended the Chicago Med School and received a PhD in neuroscience. And after that, she moved to Yale University to complete a fellowship on molecular psychiatry, and then to UT Southwestern, where she held the Ginny and John Ulick Professorship in Autism Spectrum Disorder and was a professor of neuroscience before she finally moved back to Vanderbilt University. So thank you so much for joining us today, Dr. Monteggia. How are you doing?
I'm good, thank you. I'm happy to be here.
Yes. So I think I'll start off with my first question for today, which is, could you maybe summarize your current research focus in the Monteggia lab and how exactly you think it's relevant to the field of molecular neuroscience?
Yeah, so our lab has two different research focuses. Broadly, it's focused on understanding neuropsychiatric and neurodevelopmental disorders and their potential treatment. One line of research has really focused, and I'll kind of keep it on the one line of research. One line of research is on how antidepressants work, and I'll come back to this. The second line is focused on a neurodevelopmental disorder called Rett syndrome, which is a rare disorder that affects primarily girls. And there's a gene that's been identified that causes Rett syndrome, and particularly mutations in that gene are actually what are found in the majority of Rett syndrome patients. So we've been doing a lot of work with Rett syndrome, trying to understand how this particular mutations may impact function and what happens in the brain. So that's a line of research that I'm not going to talk more about right now, unless you have questions, just for the sake of time. But the first project is actually what I mentioned before, was on how antidepressants work. And that's something that kind of strikes a chord for everyone, because everyone knows somebody that either suffers from depression or takes an antidepressant. And what we're interested in is how antidepressants work. Typical antidepressants, when they're prescribed, take a couple of weeks before you notice an effect, and they don't work in all patients. So you have this waiting time. So why do you need this waiting time? What happens? Why don't they work quickly? And so we've been doing a lot of work trying to understand how typical antidepressants work. And along the way, there was some clinical work that came out about a drug that's been around for other reasons, actually having fast antidepressant action, where when it's administered, patients within a couple of hours have a rapid antidepressant effect. And so it was really demonstrated for the first time pharmacologically that was even possible. And so with that, can we use that to really study what are the brain processes involved for an antidepressant effect? And by understanding this, it is fundamental research, but also it could have implications in terms of translational value, in terms of, possibly impacting treatment down the road.
Yeah, and if it is possible for you to tell us about this, how exactly do antidepressants work? If you could give a simplified understanding of how they do work.
So how typical antidepressants work, we really don't know. We know that they increase what's called monoaminergic transmission. It's just certain types of neurotransmitters in the brain, things like serotonin, norepinephrine, maybe even some dopamine levels. But those things happen very quickly, even though it takes a while for an antidepressant to work. And so for quite a while, the thought was, because you have this sort of lag, if you will, you take it, there's these rapid changes in these types of neurotransmitters, but yet they take a while before they really produce antidepressant effects. Is it because the brain has to do a sort of rewiring? Is there structural changes that it has to correct? Perhaps it's more spines that are an important site of action on neurons in the brain. Perhaps it's neurogenesis, new neurons in the brain. Perhaps it's circuit remodeling. So there was a lot of ideas of how this may happen because it takes time. What happened is, with this drug, which people may have heard of, is called ketamine. And the reason why people may have heard of it is at very high doses, it can be an anesthetic, even though it's not widely used in the US. At more mid-level doses, it can be an abused drug, special K. It can trigger some sort of adverse effect. What was found is that with extremely, extremely low doses of the drug, it could exert rapid antidepressant effects within a couple of hours. If it doesn't work within a couple hours, it doesn't work. And so it really provided for the first time the idea that, again, you can have a rapid effect. And it's so rapid that clearly you're not doing massive circuit remodeling, you're not doing a lot of neurogenesis or, functional spinogenesis because it's too short of a time frame. So it's possible to have an antidepressant effect without these structural changes in essence. It really suggests that there has to be something going on in terms of the function of the brain. What is ketamine tapping into so that functionally you have this rapid effect? And that's really been what the search for our research has been focused on, trying to understand that. Because if you can understand that, then are there other ways to harness that effect to treat people? Ketamine doesn't work for everyone. It works for a lot of people. Most of the studies have been focused on patients that are treatment resistant. What that means is if you are prescribed, if you are diagnosed with depression and prescribed an antidepressant, again, it takes a couple of weeks to see if it works. And if it doesn't work, then they may try you on another one. And if you fail 2 antidepressants, where, again, you don't have an antidepressant effect, you're considered treatment resistant. And there are other things they can continue to try. But it's on these patients that have been treatment resistant, which ketamine works. And so that is a huge unmet need. It doesn't work on all of them, but it works on a large percentage of them. So therefore, by understanding this, could this have broader implications for really antidepressant research?
Okay, yeah, thank you so much for explaining how it works, and it definitely makes more sense to me. And I also wanted to ask another thing about depression and what your lab works on, which is with neurotrophins. And I noticed you also focus on BDNF. And so how exactly does this trophic factor impact with depression? Like, have you seen any associations, and how exactly does that work?
So brain-derived neurotrophic factor is a member of what's called a growth factor family. And they're very important in development. We know that. That's been known for quite a while. What we were able to show very early on was that this growth factor actually, though, also has very important roles in the adult brain. It's not just due to development. If you actually manipulate its expression in the adult brain, it has an impact. And so what is it doing in the brain? And we've done a lot of work looking at this. And what we found is that rapid antidepressant effects of ketamine require BDNF to be rapidly made. And it's rapidly made because it impacts effects on synaptic transmission, on sort of neural communication in the brain. And that's required. If you don't have functional BDNF, ketamine does not have antidepressant effects. And it's true not only with ketamine's rapid effects, it's also true in terms of typical antidepressants. And so we think that BDNF and how it's made is different between how typical antidepressants work and how rapid antidepressants work. But regardless, both ways, it may have differing effects on neurotransmission in the brain, but it's required. So why is that? So that's been sort of something we've been trying to understand. The question is, we can do all of this preclinically, but does it have any clinical implication? And so in humans, the BDNF gene, there can be what's called a polymorphism, where a single nucleotide is changed. And what happens is you get a different amino acid. And so with BDNF, it's a Val66 met allele is what it's called. So it's a single nucleotide change. And if you have that, you don't release as much BDNF. And so it's not an all or nothing effect. It's sort of like less BDNF is present. So, in our models, we can actually completely delete BDNF and show it's required. In humans, it's not quite as clear cut. However, in individuals that have this polymorphism, so they don't have the full functional amount of BDNF released, it appears that they don't respond as well to antidepressants. So it's trying to understand again why that is. Why is this required? And so I think it starts to give us sort of a hook, if you will, of starting to think about what are the biological processes involved and how do they impact neural communication? What's required for someone to actually feel better?
Mhm. And so, could you kind of explain how exactly neurotrophins work? Like why maybe you think that growth factors could actually impact depression? Because, like, I mean, when I think about it, I think of growth factors as like neuroplasticity. I don't know if that's exactly true, but maybe in that there's more functional connection, which is why people have less, maybe people feel more impacted with antidepressants. Like, I'm not sure if I'm reaching out with this, but could you explain why growth factors have, like, why exactly you choose to study growth factors with depression?
Yeah, so it's actually a really good question. So when we first entered the field, we were trying to really look through the literature of what was known. And there was a lot of papers out there showing that various antidepressants, they all seem to pretty much increase BDNF expression.
Okay.
So people were saying, okay, BDNF is important. Well, maybe. Just because you increase it doesn't mean that they're required. There's many changes that are probably happening because, again, there's drugs. These drugs can have many effects, but not all of them trigger the antidepressant effect. And so one of the first set of experiments we did was we actually deleted BDNF in the brain and we looked to see what could happen and we characterized what happens. And there's a range of things that happen. But what we found is that you don't respond to antidepressants. And we did this in animal models. You don't respond to antidepressants. And that really started this whole process. People replicated it. This polymorphism, what I mentioned, was actually shown. So how do we think this plays a role? Well, we think we have a better handle in terms of ketamine. What happens is ketamine triggers this rapid antidepressant effect. And we think that it's through the fact that you are rapidly translating BDNF potentially at the synapse and at dendrites. So you rapidly translate BDNF, not just BDNF protein, but other proteins as well, that then trigger a cascade effect of inserting AMPA receptors and a rapid form of plasticity. It's not the typical learning and memory plasticity, but it's a different plasticity. And we think that if you can drive that plasticity, if you can elicit it, that might be what actually mediates the behavioral effect. So, for example, ketamine works on a specific pathway to engage rapid BDNF protein expression and then this plasticity. Are there other ways where you can engage BDNF to have this rapid protein translation effect and then trigger this plasticity, maybe independent of the pathway of ketamine? What that could be important is perhaps individuals that don't respond to ketamine, maybe they have some polymorphism or mutation along that pathway upstream of BDNF, and so you don't respond. Where then if you could maybe shunt it in a different way to get this plasticity through BDNF, then maybe you would have an antidepressant effect. So we're trying to understand what it is, but it's actually quite interesting because if you wanted to have a rapid functional effect in the brain, plasticity is one way to do it. How do you elicit it? But there's different types of plasticity. They're not all the same. And so we think that specificity is important.
Yeah, definitely. And honestly, I'm glad you covered that since, like, before this, I had the notion that plasticity, with the brain is kind of similar. And it's good to know that you need to be really specific with what you're targeting to actually get a response back. So thank you for that.
Yeah.
And I also had another question about your lab work, which is with mice, a lot of preclinical work. So in your research, you talk about conditional and inducible cell type specific knockout mice. So first of all, could you just explain what exactly that means? And then after that, maybe how you use it in your antidepressant mechanism studies.
Yeah, so what we want to do is the cleanest way to look to see if something is required is to, for example, remove the gene. So in the case of BDNF, if you just completely remove the gene through every cell in the body, animals don't survive long after birth. It's required. So that becomes sort of a challenge. Like, how do you study this then in animals? Because we want to have some working hypothesis before you move forward. More than just in vitro, as you're trying to think of really linking this to processes. So conditional knockouts allow you to use different promoters of genes to drive expression to those particular cells. So for example, we're driving expression of like a Cre recombinase, which will then, when whatever cells express that Cre recombinase will then actually delete a gene that has flanked nucleotides on it. So you sort of are crossing two animals. So if we do a conditional knockout, we can delete BDNF in particular cell types or in particular parts of the brain, for example, to try and see, is it required in those regions? Is this really brain specific? What regions, what cell types? Inducible just means that perhaps you're using some sort of pharmacological manipulation to actually then turn on Cre recombinase. So again, to get around perhaps developmental components. And again, trying to maybe more regionally target it in some way or temporally. What time do you turn it on? Maybe you want to wait until clearly in the adult and not early on because you want to rule out a developmental effect. So it's just different ways of manipulating, and in this case, deleting the gene expression of a particular gene, in this case, BDNF.
Right. And when you do use these mice, then how exactly can a mice show differences in how antidepressants work?
So antidepressants in several behavioral paradigms produce a very specific behavior.
Okay.
And so we'll take an animal and we can show that if you give it an antidepressant that works in humans, it has this sort of behavior. And so because of it, pharmaceutical companies may use these same paradigms to screen for novel antidepressants. So what we're trying to do is to use these paradigms to actually test what pathway is involved, right? What is it that's producing this behavioral effect? Obviously, depression is a very complex human disorder, but can we start to understand how antidepressants work? And hopefully this will have, translational value, clinical utility down the road.
Okay. And I also wanted to part ways and talk a little bit more about the Rett syndrome, which was another aspect of your lab. So, my first question was, since it is a genetic disorder, how exactly did you relate it to neuroscience with the synaptic faults, and how exactly could you explain maybe how that relates to the disorder exactly?
Yeah, so Rett syndrome is it's classically being called a neurodevelopmental disorder. So there's a particular gene called MECP2 that is an important factor. It's a transcriptional factor, but it can have other roles as well. And what happens is that there are mutations, again, sort of a change of a nucleotide, and it can produce different types of mutations. And those mutations can actually lead to Rett syndrome. It's sort of impairing the function of this gene. So it's not one particular mutation. It can be a range of mutations that happen. And these children, girls, because the gene is on the X chromosome, it primarily impacts girls, can be born, the first early months of development may seem normal, but as you sort of need this gene, if it's not functioning properly, you start to pick up a number of developmental delays. The children can have often they have this sort of hand-wringing type behavior. There's a range of aspects to Rett syndrome that actually have been identified. And so what we're trying to do is understand, what does this gene normally do in the brain? And what happens if you don't have it? What happens if you have one of these mutations? Where does it impact? How is it impacting? And by studying that, we start to really understand what the function of this gene is, to understand what happens when it goes awry. The role in this is, I think, important because not only in terms of how we approach Rett syndrome, but also our work on antidepressants, in that before you know what goes wrong with a disease, you kind of have to know how things normally function. It's hard to know what goes wrong unless you know what sort of a typical level of processing. For example, the role of MECP2 in the brain. What does it normally do? And then if you think about these different particular mutations, how do they impact the function? How does that perhaps contribute to the disease as you're trying to study it, and perhaps thinking about ways that you may be able to try to correct some of these deficits that have been identified?
Right. And for your research, though, is it more of a functional type thing where you look at which different brain areas are impacted by Rett syndrome? Or is it like based on gene expression and seeing which, like I noticed that you work on HGACs, which is histone
Deacetylases?
Yes,
Yes.
places, yes. And so what area of your research do you focus on then? Is it genetics, functional, or like a mix of both? Could you explain?
So we do a lot of genetic manipulations to really look at the function. What is happening in terms of the function? What's interesting is that MECP2 is a transcriptional factor, it can have other roles as well, but it's not as simple as you just have a particular mutation and then all these different genes change. It's way more complex than that. It's a very important transcription factor. And so early on, there was a lot of work trying to figure out what genes go awry if MECP2 doesn't work correctly. And can you start to correct those? And there really were not very many genes that were identified. And so there's a lot of reasons why that may be. There may be other functions of MECP2 in the brain. So what are those functions? And how can we understand them to really think about it? In terms of the function aspect, if you have specific effects on neurotransmission in the brain, and by understanding that, are there ways to correct those deficits? And if so, would that have any sort of effect on rescuing behavioral effects? That's one way of how we're thinking about it.
Okay. And also, I know there is MECP2, there's HDACs, but are there any other epigenetic factors that might be, further studied about Rett syndrome at all?
So there's a number of different genes that are being looked at. MECP2 can play a role with HDACs as well as methylation at CPG islands, which are just another sort of epigenetic mechanism, if you could. But there's many epigenetic changes that can occur. And so it's something that people are looking at. We have definitely done studies along this line, but often we're also trying to examine the impact again on function. You need some output because just because you see a change doesn't matter. And so what we're trying to do is actually measure neuronal communication and see how it's altered. Is there some specificity? Do different mutations produce similar effects? And what that may be to actually trigger the syndrome.
Okay. And actually, this leads me to my last question for today's episode. And it's pretty broad. It's just, what is one piece of actionable advice that you would give to a high schooler or an undergrad who wants to pursue neuroscience?
So neuroscience is a really, really dynamic field of study. When we think about the heart, the kidney, other organs, we know a tremendous amount about them. And we've definitely made strides in terms of the brain, but there's still so many unanswered questions about what happens in terms of normal neurodevelopment, normal brain function. And again, then kind of extending that, what goes wrong in disease processes. So it's a really dynamic time. I think a lot of different tools, approaches like genetics, as well as other approaches, have really opened the door of how we can address these questions. And so, neuroscience is really fundamentally who you are. How do you think? How do you process information? What about your senses? What you see, what you hear, your smell, all the senses, all of this is, really, it's all dependent on the brain. And so even questions that people ask, like more philosophical, what is consciousness? These are all questions that, trying to understand how people learn. How do you forget? You can't remember everything you learn. So how do you actively forget in a day? What is normal? What goes wrong in things like Alzheimer's disease, right? Where it's not just normal forgetting, but it's a disease aspect. So there's so many questions, and I think it's such a great time to actually enter the field of neuroscience because of some of these new tools and techniques. But also, I think just from some of the advances of how people are thinking, it's a very inviting field for looking for new ideas and for people that want to make a difference. And so I would really just encourage people getting exposure in labs if they have the opportunity to actually see what is neuroscience. And also, really learning more about neuroscience through coursework, really actually the study, because it is quite fascinating. So I really am excited, and I think it's really just about moving forward, just one step at a time, just keep moving forward.
Yeah, thank you so much for that answer. And I think that really wraps up our discussion for today really well. So once again, thank you so much for joining us today, Dr. Monteggia. And to everyone listening, make sure to look at her lab website, which will be on our episode description, to stay in touch with what her lab is doing and all the research work that they've been up to. And so until next time, this is Pranali signing off. Thank you.
Thank you.